Thanks everybody so late on an afternoon of this first day of the conference. Hopefully this will be of interest to you. I haven't got a rock. I'm feeling very naked without having a rock on my desk. As has been said, so a snapshot of Ireland. So we're a mid-stage clinical company. What does that actually mean? It means that through a whole heap of access to clinical and other data that we didn't pay to generate, that has been generated over well over a decade and a half, we don't go through all of the earlier stages of drug development. We're moving straight into a phase 2 clinical program to demonstrate the prevention of dengue virus in a human volunteer trial challenge in New York State in the US this calendar year. And I'll tell you the story as to how we got there and why we think that that's a really big step forward for the management of these tropical diseases for which there are no other good options. These are growing diseases. They have historically been seen as tropical diseases. We're focusing on the mosquito-borne viruses in the first instance. That's a whole host of diseases, including the one mentioned, dengue, but also other viruses that have been topical in recent years, like Zika virus, chikungunya, Japanese encephalitis and yellow fever. We have data in all of those disease states. Japanese encephalitis is interesting. JE has not been seen any further south than about here in the Australian continent historically. As recently as only about 4 months back, we had a whole string of deaths from Japanese encephalitis in mid-Victoria and South Australia. So the mosquitoes are being pushed a long way south by global warming. This year's been an extraordinary year, but if JE is reaching right down basically to Melbourne, then some of these other diseases are not going to be far behind them. So there are no good management options right now, and I'm just going to tell you what we're doing to change that. So I've talked about drug repurposing. So we have some intellectual property around our drug, which is obviously important for a drug development company. As importantly for us, we have a whole host of really key relationships that have delivered us that data and history for the drug that's been funded by other people's money, which is a great situation to be in. Part of that was funded by J&J, who originally developed this molecule as an anti-cancer drug. They got tired of putting it through clinical trials at about trial 30 and handed it over to the National Cancer Institutes, who put it through about another 15 cancer trials. Turns out very, very safe drug. Also turns out not very good cancer drug. This drug then turned up in a viral screening program undertaken by Monash University in 2017, and we picked up the global rights to that program, and that's what we're taking into the clinic for these diseases. The other key relationship we have is with the US military. We have a collaborative research and development agreement with the US Army. Why is that important? The US Army is actually quite agitated about dengue and some of these other diseases. In the Pacific Theatre in the Second World War, the US Army lost as many people to dengue as they did to bullets, and they've been fairly interested in this ever since. Unfortunately, in the 70 years since, there's been no steps forward in the control of dengue, so it's still killing as many people as it did then. So I've touched on it already, but you can go and look at this yourself. The spread of these diseases into temperate and subtropical areas is inexorable. It's not going to turn around, and it's going to continue. Dengue is really bad. If you get dengue the first time, you're likely to be hospitalised. If you get dengue the second time with the second strain, there's 4 strains of dengue, that's when you're at risk of actually dying from a syndrome called hemorrhagic fever. Again, there's been no good solution for this disease found, and we hope we have the solution for this. But there's some equally scary diseases in other parts of the world. If you remember the Brazil Olympics, a whole host of female athletes refused to go to those Olympics because of a disease called Zika virus. Zika virus is a horrific disease that's also in the same family. Basically, if a pregnant woman gets infected with Zika, the baby is born without a brain. So it's kind of a no way back disease. What about dengue itself? About half the world is at risk of coming into contact with mosquitoes that can spread dengue virus. The WHO's estimate is that about 3.9 billion people are at risk, and about a tenth of those a year, 290 million, are likely to get infected, most of them subclinical or mildly clinically, but about 10% will be hospitalised, so that's a fairly big number. We've demonstrated in the lab, and I'll come to this in a minute, that we have both therapeutic and prophylactic effects on this virus, and we act on the virus, not the host. And so any data that we've generated on the virus, kind of regardless of what setting that's in, is likely to translate into the clinic. We're not reliant on human factors, if you like, to kill the virus. We're actually acting on a viral protein itself. And then once we have an approval with the FDA and we're going through the FDA in the first instance, we will then have the ability to move from dengue into some of those other disease states using the same drug. And we're also eligible for a strange thing in the US called a priority review voucher, which I'll touch on if I have time. But in a nutshell, this is a scheme that was put in place by the FDA to encourage new therapeutic options for underserved diseases. They give you a voucher for an expedited review for a second drug when you get the first drug approved if you fall into certain categories. Weirdly and very US typically, these vouchers are freely tradable on the open market. So Big Pharma buys these, it gets them an extra 6 months of sales earlier than they would otherwise get it. And so if they've got their next $5 billion Viagra, it gets them an early 6 months of sales. So that's sort of a sweetener at the back end of our drug development process. This is dengue today, so it's a worldwide issue. This is where we're seeing outbreaks, and you'll see there's been outbreaks relatively recently in Queensland. Interestingly, for our West Australian mine owners, there's about somewhere between 300 and 400 dengue cases in the fly-in, fly-out workers in Western Australia, largely because they live in Bali. But nonetheless, there are dengue cases on a regular basis in the northwest of Australia. If we look at where WHO thinks that this is going to end up in 2050 as a result of global warming, you can see the spread of this disease has become quite significant. So you've got all of India affected, you've got a large swath of the southern US affected, you've got all of South America basically except Argentina, the whole of the top end in Australia and spreading into Europe and the Mediterranean. What are the treatment options for these diseases? With the exception of yellow fever where there is an effective vaccine, not great. Dengue fever, there is no therapeutic, in other words, no small molecule drug that's effective against it today. There's been quite extensive efforts to develop dengue vaccines. For the reason I alluded to before with the first infection, second infection problem, often dengue vaccines actually have fairly horrific side effects in that one of the major side effects in some of the dengue vaccine trials was death in the patients. So that wasn't a great outcome and most of those got suspended. There is one drug that's been approved with a really, really narrow label claim. It's approved in primary school age children with a prior history of dengue infection in a dengue infected area, which basically means primary school age kids in Puerto Rico, which is a pretty narrow label claim. And you'll see the Zika over to the side. That's a Zika baby. So drug repurposing, I've touched on it already. It was originally an anti-cancer drug, turned out not very good anti-cancer drug. Thousands and thousands of patients have been dosed with this drug. It's really safe. We've already taken all of that data before the FDA in what's called a pre-IND meeting, a pre-investigational new drug meeting. They've agreed that data is sufficient for us to progress straight into phase 2. They've agreed to the structure of our clinical trial, and that's what we're going to be moving forward and acting on in the back half of this year. We've had some manufacturing delays, which those of you who follow us may have picked up on for the first half of this year. It's been not great, but we've since solved those, and we put out an announcement about 10 days ago announcing a new supplier for our finished drug, our actual gel caps that are going into the patients. And that's a flag in the sand that now lets us roll out and flag to the markets where the rest of those milestones will fall over the back part of this calendar year and the early part of next, by which time we should be starting to disseminate phase 2 data. So what convinced the FDA that we were phase 2 ready? As I said before, we're not acting on the human, we're acting on the virus. When you're acting on a virus or a bacterium, kind of regardless of the setting, that translates pretty well through the drug development program because the environment that the bug is in doesn't really affect your ability to kill it very much. So we have a succession of studies that we inherited largely from the National Cancer Institutes that we have a license to. So they conducted some early experiments in virus in a solution in a test tube. We killed that virus and we prevented it replicating. They then moved from there into human cell culture. We killed that virus and we prevented it from replicating. That then moved to the Monash discovery process where they used a rodent model of dengue, which is a very, very, very severe model which kills 100% of rats. And we prevented death in 70% of those rats. So on the basis of all of those 4 studies, we presented that to the FDA together with our manufacturing data, and they agreed that we were